The following is the longevity news for May 2025, where I will go through a bunch of recently published papers and this time also an FDA announcement to provide an update to scientists and entrepreneurs working in the longevity industry. And after watching this, you will know about the new FDA plan to change animal testing and regulations. You will learn about a paper that asks the questions why with all this investment that we put into AI for drug discovery, we haven't discovered a bunch of drugs with AI yet. You will also learn about a new version of an aging clock and the comparison to all the other aging clocks, and of course a bunch more things. Before we start, I wanted to put out a point, and that is this is supposed to be a chill, relaxed time that we have together. Just imagine you hang out with a friend that is telling you what you learned about longevity. And if you don't like this relaxed style, I put timestamps below. There you can have them. I think you can see them on Spotify and on YouTube. And you can just jump to the paper that you are interested in. Or if you just like to listen to podcasts, just stay with me and relax. This time I actually made some tea. I made a lot, but now it's already 4 and this is green tea, and I don't want to drink green tea at 4. So I guess water it is again. But yeah, let's just have a good time together while I go through the news for this month. Who am I? My name is Elias Schlie, and I'm recording this because I wish this would exist. My goal is to make aging and death completely optional, and for that I am working incredibly hard to build a strong understanding of longevity, of the longevity field. And for this I signed up for a bunch of newsletters, a lot of newsletters. There's so many good newsletters about the longevity industry. And I was like, I'm gonna read all of them. But turns out my inbox is just full with newsletters and I'm not really reading them. But I really, really love listening to podcasts. And I would love to have a podcast where somebody just does essentially what they do in the newsletter, but put it in a chill podcast format. And because I couldn't find this, I was like, I will just try to create it myself. I want to read papers that are published about longevity anyway, and I can maybe make something really valuable. I'm still experimenting if I want to continue doing this, because as it turns out, this is a lot of work. Like, I'm spending a lot of time on trying to make this actually good, actually valuable. And yeah, I'm still seeing if I want to continue with this. Also, because this is new, it kind of feels like it's a kind of random lucky draw of papers that I chose. And over time, I will definitely increase, like my ability of choosing the best papers will definitely increase. I do think that the papers I chose this time are definitely relevant, especially the one with the FDA I found extremely interesting. But yeah, this is just a note. I'm new to this and I'm very open to feedback, so still testing out what is the best approach to deliver this longevity news. Now, let's go through the topics that I have prepared for today. The first one is the FDA release that the FDA plans to replace animal testing with a bunch of in vitro and in silico approaches that can potentially be even more accurate than what we have with animal testing right now. Extremely interesting release. The next point on our agenda is why didn't AI discover a bunch of drugs yet? This was published by, co-authored by Alex Zhavoronkov, and yeah, he is definitely in the game with in silico medicine, using AI for drug discovery. And this is just the question that, we invested so much money into AI drug discovery, but it's not that every week we hear, oh, there's a new AI drug and another new AI drug that is approved now. Of course we have here some news about this, but there's nothing on the market yet. And they ask why this is and how we can actually make it work, that long term we will find this. The third point is about, or this third paper is about Lineage 2, which is the new version of an old aging clock that was called Lineage before, now Lineage 2. The paper was published by Fong et al. And it's called Lineage 2, Providing Actionable Insights and Benchmarking with Epigenetic Clocks, where it compares different epigenetic clocks and just puts out this new clock that I will present. The next two things are two events that I thought are very interesting, and I just wanted to mention them here. It's one, the Frontier Tower, which is like a house that was bought for innovators in San Francisco, a big multi-story tower where they will also have a floor just dedicated to longevity. I will talk a bit more about this when it's the turn. And the other thing, Vitalist Bay, which is a big meetup, a giant longevity pop-up city in Berkeley, California, so very close also to San Francisco. And I know a bunch of people there, and I think what I hear, it is amazing, and I wanted to mention this here as well. So let's start with the first one. The FDA plans to replace animal testing, and they put this out in this press release, or in this, I read through their roadmap paper where they introduce why they want to replace animal testing and what exactly they want to replace this with and how they want to change their policies. And I'm going to present a short version of what exactly they want to replace animal testing with and what their motivation for this is. So they want to replace animal testing with something they call new approach methodologies, short NAMs, and those include organ-on-a-chip systems and a lot of computational modeling and a bunch of other things. I'm going to go into detail into what those things are that they will consider to replace animal testing with. But first of all, why do they actually want to replace animal testing? And the thing that they mention over and over is that animal testing is actually not as accurate as one might think. So 90% of drugs that are safe in animals don't get FDA approval. And they put out a very striking example, which is TGN 1412, which is a compound that was tested safe in rodents, in rabbits, in dogs, and in non-human primates. But when they administered it to the first patient, it turned out to have a life-threatening immune response of those first patients that got it, and which was really dangerous. And this might have been prohibited if there were more accurate modalities to test drugs. And it just shows that animal testing isn't as reliable as one might think. So they cite a lot of papers on this. Also, another reason why they want to replace animal testing is that, the ethical reason that, if we have the ability to test a drug in vitro or in silico even, we don't have to have those animals, and there's potentially less suffering. The third reason they name is the potential cost-saving advantage, that especially things like computations can be so much cheaper than animal testing, which is incredibly expensive and takes super long. Actually, that also a point, just the time it takes. Those are just the reasons why they say they want to replace animal testing within the next 5 years. They want to start with monoclonal antibodies. And why do they want to start with specifically this? Monoclonal antibodies are related to the immune system, and the fact is that the immune system is incredibly different between species. So testing immune system drugs, there's such big differences between species that is very unreliable what we get right now. And the standard right now for monoclonal antibodies is to do primate experiments, which are incredibly expensive and take super long, and on top are very unreliable. So replacing this with the proper new approach methodologies is the first step they want to do, but over time they want to replace more and more of the stuff that we use animal testing right now with those new approach methodologies. And now let's go through them. Like, what are those NAMs? What are the different things that they want to replace animal testing with? First of all, there is the in vitro human-derived system. So it's like the first bucket that they name. And this bucket includes microphysiological systems and organoids. I'm going to go through them one by one. The first is microphysiological systems, also known as organ-on-a-chip systems. And those are essentially like highly controlled environments with human cells inside. So you have to imagine like a little chip on which human cells are put, and then there is like different flow between different chambers, and the chambers are arranged in different ways to model actual human organs. And the advantage of this is that it actually uses human cells. So there is more resemblance to what is human than in animals. So in a mouse, it's mouse cells, it's not human cells. The problem is that it isn't actually a full organism. It's just some cells on a little plate, essentially, like a more complex plate, a chip. But yeah, this is just like way less complexity because of that, which is kind of a contra, but still, these microphysiological systems, in previous studies, they show to predict human response very well and partially even better than animal tests, which is very promising, and this is why the FDA decided to include those microphysiological systems as a possible replacement for animal tests. Another like maybe counter-argument against microphysiological systems is that they lack the inter-organ interaction. But the cool thing is this can be solved, or people are already trying to solve this by building multiple organ multiphysiological systems, so that you essentially have connected, let's say you have a chip for the liver and then you have a chip for the heart, and then you can kind of put them together, that they share the same circulatory system, and you can see how they influence each other, which can be extremely valuable. And if you can think about this in the future, like we can maybe build like really intricate, complicated systems that are really accurate in tracking what the human response to a particular drug might be, which is extremely promising, and also I think really cool that the FDA is looking into this to replace animal testing. The second thing of this first bucket in vitro human-derived systems is organoids, and it has essentially the same pros and cons as microphysiological systems. Organoids are self-organizing cell cultures that model human tissue structure and function, so it's less controlled than those microphysiological systems, but in a sense it's still relatively similar. It's in vitro testing, which is also promising, but I think a bit longer already existed than microphysiological systems. The next thing that they want to, the next big bucket they are naming is in silico new approach methodologies, so things related to computation. And I wanted to go into those in detail. They're all listed in the FDA paper, and also there they just mentioned them, so you can look them up in more detail if you want to actually get drug approval from the FDA. But the cool thing is about that is that drug testing can be, computation can be incredibly cheap and fast, and if you can use that for drug testing, it might be able to speed it up immensely. The three things they mention is, first of all, physiological-based pharmacokinetic modeling, which is essentially like an in silico drug testing approach that uses mathematical simulation of the drug ADME, so the drug, how it's absorbed, how it's digested, how it's metabolized and excreted. I think I'm not fully sure, but like the full interaction of how the drug moves through the body, what happens, and this is modeled by those physiological-based pharmacokinetic modeling. Very interesting. The second thing they mention is AI drug testing. So essentially it's just using this vast amount of toxicological data that we have to train an AI on predicting if a certain drug will cause a maybe immune response or toxic, it's going to be toxic. And what they suggest doing, or what this approach is that they want to bring forward, is to have this large-scale AI models that predict from the structure and different features of a new drug how the person will react, if it's going to be toxic or not. And yeah, this is how we can test very quickly new drugs, if how much promise they show. The last in silico new approach methodology that they propose are the QSP models, so quantitative systems pharmacology models, which simulate how a drug interacts with the human biological network. And essentially you can think about it like a virtual human, so you can test drugs this way. All still new technologies that are worked on, but quite promising. And FDA is not saying, I'm also going to go in detail later, but FDA is not saying that if you have one of those, if you say, oh, the AI predicts that this drug is safe, like you're good to go and can put it into humans without any other tests. But this is essentially their approach is like, yeah, slowly roll this out. If you have multiple, if you show multiple, you get a waiver on your animal testing, so you can do less animal testing. And yeah, just over time learn more and more about the safety of those approaches as they are developed, and then eventually completely replace animal testing. The third bucket, just other modalities that they suggest, is, for example, ex vivo human tissue drug testing. So taking human tissue, actual real human tissue out of a human and testing how it interacts with drugs. The second thing they mention is cell-based drug screening. So having a lot of single cells. Actually, last month when I did the longevity news, I made one about large-scale, how's it called? Never mind. Yeast, exactly. They had a bunch of yeast and tested how yeast aging. But what they would do is like actually human cells, and then large scale to see how human cells react to the drugs. So that would be like a small number or even single cell testing, but still can have value. Third thing, PET microdosing. So a PET is essentially that you give this drug, you give it you kind of make it radioactive, and then you take a microdose of this drug, and that way you can see, if you go in this PET scan, that it can detect where this radioactive substance travels in your body. And that way you can see how the drug is absorbed in your body. So if you take this microdose of radioactive drug, so the drug made radioactive, it will go into certain parts of your body, and that way we can learn about how the drug distributes inside the body and learn about the drug. This way without big risk, because first of all, radioactivity, if it's a very, very small dose, it's not such a big of a problem. And because it's microdosing, you really take just like a minimal dose of this drug, there is way less of a risk of something going wrong. The last thing they mention is humanized transgenic animal testing. So essentially you take you genetically engineer, for example, a mouse or like an animal, to have genes more similar to humans, and to just show characteristics that are more similar to humans, and then test on them. So have animal models that are kind of like humans, but test on them, which is still not perfectly validated or anything, but which is definitely something worth looking into. Now, the policy changes they want to bring through is, first of all, that they, like, a really cool thing, additionally to replacing this with NAMs, is that they want to start accepting also data from other countries. So if there's another country where the regulations are similarly strict, then, and in this country a drug is already approved, for now the rule was that if you want to get your drug approved in America, you have to go to the FDA and you have to go through the studies again. And now they want to start accepting data from other countries, so you don't have to do everything again just because you also want to have it approved in America. Another policy change they wanted to make is that if you use those NAMs for now, they will decrease or at some point hopefully completely eradicate the need for animal testing. And yeah, right now the more the better. The more NAMs that you can show as having a positive indication for your use, the more likely it is that they will waiver some animal testing requirements or decrease the amount of animal testing that you have to do. The last thing is that they want to do different trials and compare how the different approaches and how strict they are influences the success rate of the drugs they put out. So make different experiments of having intensive animal testing together with new approach methodologies, or all the variations, like one of this without the other, other way around, a lot of one with a little of the other, and just test how this influences success and safety of the drugs. Also, what they want to do is they want to build a big master drug toxicology data set, because there's none that exists right now, or at least there are some that exist, but those are specific to some countries or specific to some specific collaborations. And what they want to do is they want to round up all this data and create this database where different compounds are rated with features and their toxicology data, which can then be used for example, training an AI model on predicting the toxicology of a new compound that you came up with. So the next paper was written by Dominika Wilczok and Alex Zhavoronkov. Alex is the CEO and founder of In Silico Medicine, and they use a lot of AI for drug discovery. And this is exactly what this paper is about. This paper is about progress, pitfalls, and impact of AI-driven clinical trials. That is the name of the paper. And the question they ask is that we have put so much money into AI for drug discovery in the last around 10 years, but there are not that many drugs yet. Essentially, there are no really AI-discovered drugs on the market. They completely passed all the safety testing yet. And this is curious because there's so much money, there's so much promise that the AI is going to solve everything, but there's nothing to show for it right now. Of course, there are drugs that advanced, but nothing really on the market right now. And this is what the question why this is what they address in this paper. But before they address this, they lay out like the three different strategies that the companies that use AI for drug discovery use in the moment. So there's three different strategies how to use AI in drug discovery. The first of those strategies is the strategy of repurposing or in-licensing drugs based on AI hypotheses. So the advantage of this is if you take a drug that is already approved for another target, another implication, and you figure out that it's also good for your implication, you're incredibly fast at phase number 2. So phase 2. That means that you don't have to go through this, what we just talked about, all this animal testing, toxicology studies, because this already has been done when the drug was approved for its primary first implication. The problem is though that this has been tried a lot of times, and incidentally, it failed most of the times. There wasn't just the target choice was bad, they chose the wrong target, and in the end it didn't work out. So yeah, many failures on that approach. Another benefit for this approach is that you have an extremely low chemistry risk, because yeah, the chemistry already exists, so there's not a big risk that your drug doesn't really work, doesn't get absorbed properly by the body, because all of that has been tested and figured out when it was approved for it, or when it was through its use for the first primary implication of this drug. The second approach to AI drug discovery is to design new entities for established targets. So now it's the other way around, not an old entity for a new target, it's a new entity for an old target. So this is then where AI comes in. So you have this target that is already kind of validated to be implicated in a specific disease, and now you use AI to create a new drug that targets this specific target. And there you have a very low target choice risk because the target is already validated, but you have a high chemistry risk, other way than in the one mentioned before. And the problem is there's also a lot of competition. So for established targets, there are a lot of companies at the same time working on them. And yeah, it's just the risk that somebody else just arrives there earlier or arrives there cheaper with a cheaper drug or something. And then that's would create a problem, or like all your work would go to vain. The third approach is to design novel targets for novel molecules for novel targets, and this is where the whole AI end-to-end approach. But this obviously has both of the problems, like extremely high target choice risk and also high, or as they say in the paper, moderate, but also chemistry risk that something can go wrong in all the parts because you're completely new. Of course, this way you don't have any competitors and you don't have to be afraid of that, and you don't have to pay for repurposing a drug, but at the same time, there's a lot of risk involved in this approach. And now to why exactly we haven't discovered a lot of AI drugs yet. So the challenges and opportunities that AI-powered drug discovery right now has, especially like the challenges. And one of the biggest ones that they mention is that there's this huge lack of benchmarks at the moment. So there's not really a good way to compare the time and cost of different drug discovery approaches, because companies just don't publish their data on how long it took them or how much it cost them to get a certain drug to market, or all of those failures even that they have. Like the success rate is nothing that companies usually publish. And if you want to integrate new AI algorithms into your pipeline, it would be incredibly valuable for other companies to publish this, or so you have something to benchmark against, to compare against. If your AI improves maybe the accuracy, the success rate, or the speed, the cost, anything of those benchmarks. But the fact that people don't share this makes it hard to compare, and it creates like this focus on this bias towards focusing on like the shiny new news that is it sounds cool, like, oh, there's this new approach, it sounds so cool, let's try this. Like, of course it's not that bad, but instead of like really focusing on the hard metrics, and there's just not enough analysts working on this benchmark problem, and not enough published. Another problem they point out is that there is an incredibly fragmented focus. So often there are companies that focus only on a subset of the drug discovery process. So for example, a company that focuses on the chemistry part, but they don't focus on their clinical trial program, and therefore they fail, because it's just not a holistic approach of getting the drug fully to market. It's just like the first step, and then they fail. Another problem is what they call the expert gap, and that is that there are a lot of expert drug hunters who are way too careful with AI recommendations, and therefore they don't use the full potential that AI has. But on the other hand, there are also the AI experts who are way not careful enough, and they just blindly follow. Of course, not blindly, over-exaggerating, but I'm not. So they follow the AI advice while not being careful enough, and therefore they fail. And yeah, just bringing those two groups together and finding the right balance between being careful with AI recommendations but not too careful that you in the end don't really end up using them, would be a great unlock and a big opportunity, but also a big reason why it's not working that well right now. Another problem they point out is the curious fact that there's extremely low collaboration success between AI companies and pharma. So there are a lot of collaborations between new startups, AI startups, and pharma, but the success historically is extremely low. Another point they mention is that there is this misplaced innovation, that many things try to solve previously unsolved problems and don't focus on things that on improving things that we already know that work. So everybody's always, or many people are trying to go to this new thing, to this thing that we don't know yet how to do, and are like, okay, I'm going to solve this problem that nobody has solved before, instead of just looking at, okay, this is what works right now, and I'm going to make this 10% better. I'm going to make this 50% better. I'm going to make this cheaper, and all of that would lead to higher success rate and more progress overall, because all those new approaches are just extremely hard and improving something might be a better thing. And they don't say that you shouldn't try new things, but just that this balance is kind of off, that people focus too much on creating new things than on improving existing structures. Another big problem is that those models, the AI models, are extremely poorly validated, and this is just, they have to be tested more rigorously before people start trusting them, because if you just trust them without good testing, then it's often what we saw happening, it goes wrong, which it did so often. Another problem is that the public data we have is super disorganized, and it's just a mess that you have to search for data in every place, and that way it is hard to really train your model on the highest quality data, and because it's just such a big effort to find all the data and to format it in the right way that is kind of useful and you don't have 5 different data sets that don't really overlap, that overlap in some things, and then you can't train your model on all of them. You have to work with the data to make it to use all of the data, which is a lot of effort, and which is right now everybody has to do it again and again and again that wants to use this data. To conclude, I also have the source here. Is that for people listening, I have the source on the video. For people watching, I think you can watch the video on Spotify or on YouTube. Their conclusion is that AI right now is often used in automating routine tasks, which is nice, which is tasks like medical writing, regulatory submissions, data analysis, but all of this won't lead to crazy breakthroughs. Would be the real uses where there might be incredible benefit is to help AI to integrate what we already have in preclinical knowledge into decisions. So similar what I talked right now about before this about with the FDA, that using AI to validate drugs before even using them, to help integrate historical clinical data into your decisions. And another use would be maybe to identify good biomarkers using artificial intelligence, and also possibly suggesting combinations of different therapies to find what works together and might even increase the effectiveness of each other. So the next paper is about lineage2, which is a new version of an old biological aging clock. The old version was called lineage, just lineage, the new version lineage2. Both of them, all the new version, were trained on the National Health and Nutrition Examination Survey, NHANES 4, in quite a big range, I think from 1980 something till 2000. And after a bunch of feedback that was given by people that were using Lineage, they updated it to Lineage 2. And in this paper, they present the changes they made to the clock, and at the same time also compare to different clocks and kind of benchmark different clocks against each other on different metrics and compare their ability to do what biological aging clocks do, and that is to predict biological age. In the paper, they define biological age as age at which a subject of the age at which subjects of a reference cohort have the same risk of age-dependent disease as all cause mortality as the subject in question. Sorry for bad reading. But essentially what that means is that biological age, you probably know about it, but it's if I track the mortality, all-cause mortality and rate and age-dependent disease risk of people of all ages, and then I measure myself, and at the point where I have the same level of, at any point I have a certain level of age-dependent disease risk and risk of all-cause mortality, and this level is supposed to be matched to this cohort. So let's say people that are 25 have a certain level of age of those risks, and I'm right now at exactly the same level, then I would have a biological age of 25. Or if you say significantly older, if you're like very short before dying, you have one of 90 or something. So that's the definition they use, and that's the function of all those aging clocks that are presented in this paper and compared in this paper. So lineage. Lineage is a specific aging clock that uses principal component analysis, PCA. What that means is that they take this high dimensional vector, this high dimensional collection of different markers that you can get, like you can see your respiratory rate, you can see different, I'm not sure if they have respiratory rate in there, you can see different blood markers, you can have a bunch of different markers that flow into this clock, and through PCA they break this high dimensional space down. They look at this high dimensional space for of this many different measurements where a bunch of them are related. They look at this and reduce the dimensionality so that you create from this large number of correlated variables a small number of uncorrelated variables. And then you have this small number of uncorrelated variables which kind of say the same amount or carry the same amount of information but with just less variables. And from those, like the cool thing about this is what they say is it's kind of more interpretable, because then this is fed into another algorithm which then predicts your biological age, so those principal components. But those principal components itself have you can kind of analyze them. And what they also did in this paper is that they showed different heat maps. I didn't put it in the presentation this time. Next time when there's something like this I will. But later I have figures for the paper in the presentation. So in heat maps you can see what the different principal components, so the different things that they got out, what they're correlated with. So you can see, okay, there's maybe a principal component that is mainly correlated with cardiovascular disease, or there's one that is mainly correlated with diabetes, or type 2 diabetes, or obesity, or whatever. And of course it's not as clear cut, and that's the bit of the complicated thing with those principal components, because we don't decide what the principal components are about. The principal components kind of like decided, the algorithm decides it, because the algorithm is about finding the most efficient way to compress this data into those principal components. But yeah, this is the advantage of the lineage aging clock that uses these principal components, which can be used to, for better interpretability. Wow, what a day. The limitations of this aging clock are that it mainly uses linear techniques, and the problem with linear technique is that you can't really learn U-shaped response patterns. So where there's a golden mean, like there's of most things, where there's not just one thing that is always better and one thing that is always worse, but whereas there's like a middle ground, and that's quite hard with linear techniques. And also it's worse at distinguishing between age-dependent disease and actual age. And what they say, like future with generative AI and neural networks, where there are already a bunch of deep aging clocks based like on deep neural networks, might be better than this, because they are able to actually capture those relationships. But at the same time, those are way harder to properly interpret, and so there's this trade-off that you have to make. And they just point this out in this paper as being a limitation of their model, but at the same time, it's working really good. I will go through the results that they have and show this. Oh yeah, here on the presentation now also says that there's PCA involved. By the way, if you're listening to the presentation, I say anything, there's nothing you're missing, it's okay. But if you want to watch it, I think I have a very beautiful screen with a very beautiful presentation. But yeah, however you want. On Spotify and YouTube, you can look at it. Now, the problem that, or at least let's say the critique that was put out to those for lineage 1 or lineage no number, what they try to then improve upon with lineage 2 was, one, that first it didn't handle outliers quite well, and there were like some threshold effects. Also, there were some measurements, like for example, fibrinogen. There's another one where it's super hard if you always only read things to say them for the first time on the podcast. I'm still waiting to have 100 biology, longevity scientists, friends around me that always talk with me about fibrinogen, and so I know how to properly say it and I don't have to make it up on the fly while doing a longevity news podcast. But you know, that's what happens. So this is a measurement that takes like some extra steps to measure, and the clinics that used this aging clock were asking them to remove it because it's extra effort, and if you can kind of get rid of this makes it way easier, and also a bunch of other parameters that are not as necessary, let's say. Another ask that was made is to improve the interpretabilities of the different principal components and provide strategies to optimize the different principal components to lower the biological age of the patient. So what did they change? Now they only include a total 60 different biomarkers that you have to put into this clock, and like without this serum fibrinogen, without, wow, this is going to be so hard. I'm going to go through it now. Gamma glutamyl transferase, total cholesterol, high density lipoprotein, HDL, and triglycerides. Okay, those were not so hard. Those I know. So they removed those measurements that were not helping that much and that were making it unnecessarily complicated to make this clock, and now it's only 60 biomarkers that are left. Also, what they did is they improved outliers and decreased like this threshold effects that I was talking about by capping outliers at 6 standard deviations. That means people that were really far from the mean at any kind of measurement, like more than 6 standard deviations, they just put them at the point of 6 standard deviations. And whenever now you have, let's say you want to measure your age with this clock, or you want to measure a patient's age with this clock, you can put at this 6 standard deviations level, or it automatically puts it there. They also log transformed some parameters. This is like a common technique if you have parameters that go like extremely high in the higher ends, and with a logarithmic transformation, this is not as extreme. Like, I think you probably know how logarithmic transformations work, and I don't have to explain them here on the Longevity News Science podcast. So as I already said, less parameters and biomarkers needed, better handling of outliers and threshold effects. That is what they changed. Now, comparison between the different clocks. They compared a bunch of different clocks on the data set that they use on the last, I think, 2 years of this. And how was that called again? The data set was called the NHANES 4, so National Health and Nutrition Examination Study. And for that they used 2001 and 2002 data to compare different algorithms on. And if you're watching this, you can now see the graphs that they published in the paper. I will also explain them if you're listening to this. So as a baseline, you can see two, like one, the chronological age, that's the white one on the most left in this area, is chronological age, and the blue one is something they call crystal age, which is supposed to be like the perfect aging clock, like a theoretical aging clock that can perfectly forecast patient's death. And that can be used as a theoretical unachievable, but that is what you're kind of aiming for to have in aging clocks, to build in aging clocks. So it's like the direction you want to go. This is as good as you can get. And this first 3 graphs, they show the comparison of, on the x-axis you have the survival probability of people, and on the y-axis you have the survival time of people. And then you have, for each clock, you have kind of 2 lines, a straight line and a dotted line. And the straight line is describes how how well the clock predicts, or not that. The straight line shows the upper quartile. So the clocks, they take the data set of people and put them in the upper and the lower quartile. They rank them. What they do first is they rank them. So everybody gets the biological age, and there are people that are older and people that are younger. And then you take the highest 25% and the lowest 25% for those graphs. And the highest 25% is what is the upper straight lines and how long they live. So it should be as high as possible. This is the theoretical perfect performances that you see that for very long nobody dies, and at some point some people die. And then you also take the lowest 25%, so those should be the people that die first. And there you can also see the theoretical perfect performance and how it compares with different clocks. And as you can see here, like for example, like Horwath age by Steve Horwath, a very known clock. It's one of the, I think, epigenetic clock. Actually doesn't perform as well if you compare it to chronological age. It's like nearly at the same level as chronological age. The same with Hannum age. And then here in the B plot, you can see the orange line is actually lineage 2, and you can see the performance is quite good, like way better than most of the other clocks. Here it's compared to chronological age, the white line, which is the lowest, and pheno age, clinical, which is kind of in the middle between the two. And then in the last one, you can again see like different clocks compared, and also there lineage 2 kind of stands out as quite a good clock. And yeah, this was done on the 65 to 75 year old cohort in this NHANES 4 dataset. And yeah, just as I already explained, upper and lower quartile compared. Another graph here is the trade-off between specificity and sensitivity. So if you can imagine of predicting people's death in the next 20 years. So if you imagine the aging clock predicts age of people, and you say kind of the oldest, this many people I will classify as dying in the next 20 years, and the other ones I will not. And then you compare how well this matches reality, and on there you can obviously you can change the threshold. So you can say, I'm gonna say everybody over the age of 25 will die, and then it's very likely that you have nearly everybody that will die in this data set. In this, so you you like perfectly have all the people that will die predicted as already dead, as dying, but this is not very helpful if you just say everybody will die in the next 20 years. So the same way, other way around, if you say nobody will die, you will be right with everybody that doesn't die, but not with the other half. So there's kind of a trade-off of where you set this boundary, and this is what they show in this graph. And there also you can see that lineage 2 is extremely good, high up. Like another thing that is quite close is actually the old lineage algorithm, and I think also PhenoH Clinical is relatively close up there, but yeah, all of them quite good. Also GrimH2, very good aging clock that is compared here. Then another comparison where you have different clocks. Let me see. You have on the x-axis here, you have the cognitive score and the gait speed of different people. Yes, perfect. On the x-axis, you have the cognitive score and gait speed of people, and you also, again, you take the highest and lowest predicted 25% of the data set, and then you compare them. So the people that are predicted to be the youngest should have higher cognitive scores and higher gait speed than the people that are predicted to be the lowest. And the same with cognitive score and gait speed. And here, again, you can see that Haworth H performs quite poorly, like P of 0.12 for cognitive score, which is not significant, and for gait speed even worse, 0.06. And then if you compare this with lineage 2, which has 1.13 to e to the power of minus 13 times 10 to the power of minus 13, so like a lot of zeros, 13 zeros, and then only there comes the 1, and for gait speed also 11 zeros, and then there comes the 1, so extremely good. And yeah, but also GrimH2 and Dundon Pace are both statistically significant, if only with 7, 4, 4, and 5 zeros, but still, like, this is enough zeros to definitely be statistically significant. Although the best performance is lineage 2, but also you have to say it's their paper, so of course they show where their model shines. And yeah. Last comparison, then we're done with those comparisons, is the comparison for average clock age and the ability to perform. So they split it up into kind of two groups. So with those measurements, there was also a questionnaire, and people could put in this questionnaire if they are able to work or if they're not able to work. And then you can, of course, put them into two groups, and you have the one group of people able to work and able to perform daily activities and able to do everything without needing assistance. You can compare them with the people that don't have this ability to do all that by themselves, and then you predict the age of all those people. And of course, the people that are not able to do their daily tasks alone, they should definitely get an older age than the people that are able to do the daily tasks at home. And again, you can see here Haworth H performs quite poorly, 0.11, 0.68, and 0.99, which is like not statistically significant at all. And yeah, compared to that, Lin H2 again performs really well, 12 zeros, 6 zeros, and 0.02. And the last one, Grim H2, also performs quite good on ability to work with 9 zeros, on daily tasks 0.02, and on the last one also like kind of similar to daily tasks, not statistically significant, but in general like yeah, quite a good performance. The general one last general finding that they made in this paper by comparing the study is that the clocks that are trained on survival and function predict mortality better than clocks that are trained on chronological age. But this is also quite obvious, I think, because when you train a clock on survival and then you test it on survival, of course it works better than when you train the clock on age and you test it on survival. So it's not so surprising, but I still wanted to point it out. And yeah, let's go next paper. And what I realized now is that there actually is the last paper, or that was the last paper, because now there's only 2 other kind of events that I wanted to present, which I think are really cool and where I know a bunch of people, and I was like, yeah, that's definitely news. That's 2 cool things. The first thing is called Frontier Tower, which is a building in San Francisco that was bought by a couple of people, and this tower is thought to have like different the different floors to be themed about different things. And there's going to be a longevity floor. Here is their website, Frontier Tower. You can just search it if you want to see it yourself. So they will have a co-working and library floor. They will have a human coordination and decentralized science floor, some crypto, decentralized science, Ethereum, and they will have floor 11 will be longevity and health. They will have artificial intelligence, machine learning floor, biotech, neuroscience, brain-computer interfaces, makerspace, arts and music, wellness and gym, deep tech and robotics, library, co-working spaces, offices, event spaces, and also co-living. And what I heard is that the vibe is just amazing and that people are having a really, really good time. And this is like a place, I think, bring people together that want to work on ambitious, cool things, and not only longevity, also all the other things. Like I would love to be on the AI floor. I would love to see what people on the deep tech floor do. BCIs, all super interesting things that I think it's just a cool project. And like this is the place where I would love to live. Like being surrounded by people that are motivated the way I am and that want to like create great things. That, yeah, just good vibes in San Francisco. So if you're there, might be worth a visit. Or actually also, I think they're still searching for people that like kind of help organize the longevity community or different communities. So if you feel called to that, amazing. Another really cool thing is something that is called Vitalist Bay. And this is one of the biggest longevity events in ever, maybe. It started on April 4th and will end on May 29th. And it's created by a group called Vitalism, which is like a moral philosophy that states that life is good and death is bad, and just also that aging is the first. I think aging is like the primary agent of death, and that we should do something about that. And I think that's really cool. That's a good mission. I'm kind of following that. I'm also in this community, and they created this big longevity pop-up city. And I already, as you might know, I made a video about it. I helped to organize a longevity pop-up city in Berlin called Zeller City, and that was very cool. It was a very cool experience. I met so many amazing people there. And yeah, now this is like that, but even bigger on steroids. It's in Berkeley, right next to the Golden Gate Bridge, or near that. So also next to SF. And yeah, it's just the biggest longevity event in the world. They had speakers like Brian Johnson, Linus Peterson, the RetroBio people. RetroBio, amazing company. I would really love to work for them. Are there. Patrick Linden, my Zala City crew, the people I know from Zala City, Lawrence I and Victoria Forrest are there. And yeah, also what I'm hearing, it's just an amazing vibe. A bunch of cool people. You can see here. I'm now on the website for people listening, just scrolling around a little bit. That there are like workshops with many speakers. They have like this campus where you can do blood testing, DEXA scans, cold plunge, sauna, workout, VO2 max test, all of that. But all that brought together to have like this community that is united under this goal to make death optional, work on longevity, and also, of course, do all this wellness, healthy living stuff, but also actually fix it. And I think this is just amazing. It's such a cool experience to live with people. Like if I think about my experience in Zala City in Berlin, it's just so much more intimate than being on a conference where you're just like, oh, hey, my name is Elias and this is my LinkedIn, or what is your LinkedIn, or what do you do? And in a place like this, it's just way more personal, and you can build true friendships that just last. So if you're there, check it out. Yeah, some more here, maybe some picture of the speakers, maybe you know some of them. In the end, I think it's just a really cool event, and that's what I wanted to present. And with that, we went through the whole thing. That is the longevity news for May 2025. Thank you very much for listening, and see you in the next one. Bye bye.